Hey guys, welcome back to the channel. So in a video that I recorded last month, I went through the latest medical AI research papers, read those for the first time, and just shared some initial thoughts about the papers. And I shared that video on this channel. It got more views than I thought it would, and I had a few people reach out to me and say that they found it interesting and would be interested in seeing more. So this is a follow-up to that video, and what I'll be doing in this video is I'll be reading all of the interesting medical AI research papers that were published in the month of February, and I'm going to give myself 1 hour to do it, and I'm going to be setting myself a timer. And throughout I'm going to be explaining the papers and explaining some of the interesting concepts about them, so hopefully this will be interesting or useful to some of the people watching this video. And I'm aware also that this is probably not the most digestible format for people who want to understand the latest medical AI research, and something that I'm experimenting with at the moment is the best way to record and share this information. So what I'm going to do with this video is, as well as just recording it as a hopefully under 1 hour video of me reading all these papers, I'm then going to cut up different sections and try and group them in terms of themes and release videos where I discuss those themes in more detail. So if you're less interested in watching a long form video of me talking about all these different papers without an obvious structure other than the fact that it's in time sequence, then you may not want to watch the whole of this video. There will be timestamps in the description below, but you might also want to look out for future videos where I will be grouping this by themes and trying to bring together some of the different papers that have been published and some of the general themes that they raise. But with that being said, let's now commence a reading of all the interesting medical AI research papers that were published in the month of February. So 1 hour starting now. And the website that I'm going to use for this is a website called drpenguin.com, which is a super helpful website. Essentially, they send out a newsletter every week showing the latest medical AI research papers. So I'm going to go back to the first week of February, which is actually not this newsletter here. I think it will be week 34. No, sorry, that's January. So that will be week 38. So this is the first newsletter of February, and there were 4 papers published in this week. There was this paper using convolutional neural networks to develop a general methodology of rapid and autonomous artificial crystal symmetry. Okay, so one caveat for this series is I'm going to skip through the papers which are very much kind of like biochemistry and protein folding and these sorts of papers because they're all outside my area of expertise, so I can't really make valuable comments on them. Okay. So biomarker of patient outcome for primary colorectal cancer resection by analyzing histology scans, applying deep learning to echocardiography, and using a UNet to detect infarction in patients with stroke. Okay, so let's look at this first paper, see if we have access to this. Just going to move this a little bit as well. Okay, so we don't have access to the full paper, but I'm just going to read the abstract and glean some interesting things about this. So, we need markers to help stratify people with early stage colorectal cancer. So they train CNNs, which are networks specialized at analyzing images. And there's this idea popping up now of kind of biomarkers, a new type of biomarker. So traditionally, as I understand, biomarkers were commonly typically associated with blood tests. So having markers in your blood of different levels of proteins or other molecules. Whereas now, with AI, there's all these different types of biomarkers you could have. So for example, if you had a CT scan and you measured the size of the liver in an automated way, or it doesn't have to be automated, but an automated way can make it more easy, then that can be a biomarker, the size of the liver detected by a CNN, although whether or not that would be that useful is up for debate. It could be. So here they're making biomarkers by analyzing these histology slides and using that to predict outcome. So what sorts of things they're looking for on the slides? Let's have a look if they say. Fine, so biomarkers with a good hazard ratio in the analysis of the cohorts. Okay. Have I missed something? Are they saying what the biomarker is, or are they just saying it's features on these slides? I'm not too sure. Fine, but that's interesting. I think one of the reasons that we see histology-based deep learning pop up all the time is because deep learning is pretty good at analyzing. It's pretty good at analyzing images, and it's pretty good at concrete steps of the kind of clinical pathway. And in histology, it is very much within a confined environment. The task is kind of a specific task to be done, which is you have an image of a slide and you're analyzing that to determine what features are on there that's relevant for disease in terms of the presence or absence of disease and in terms of the prognostication of that disease. So that's why it's pretty useful. It's pretty contained. So just having a deep learning algorithm applied to that can be quite useful. Whereas some of the other tasks that you might have, such as let's say chest X-ray interpretation, there's perhaps a little bit more to it. It's a little bit less contained because you typically will analyze a chest X-ray in combination with different features about the patient's clinical presentation, the symptoms, the examination findings, which I guess still is the case in histology, but I think less so. At least that's my impression. So then we have a paper about looking at analyzing echocardiograms. And again, we've got deep learning. So a lot of the machine learning papers that we're seeing now are these deep learning papers, typically convolutional neural networks. I haven't yet seen, I've seen a few RNN papers, but the vast majority are deep learning, are CNN papers. So analyzing echocardiograms. EchoNet, I like that name. Okay, cool. And we have got the whole paper here as well. So basically, what they're doing here is they're looking at echocardiogram images and using a CNN to analyze that. So looking at the sizes of the chambers, the sizes of the muscles, and trying to detect the presence of, let's say, hypertrophy or an enlarged atrium against a predefined threshold, I guess. Or not predefined, but against a threshold that's been defined, and then they'll have given some labeled examples of enlarged atrium and not enlarged atrium, and then this network is able to achieve pretty good performance scores, pretty good AUC scores to distinguish between those different types of pathology being present or absent. And that's quite cool because it can support technicians who are doing these kind of scans. In theory, perhaps you could have somebody who's less experienced at doing echocardiograms and have this giving kind of a supplementary analysis. I think also one of the challenging things about echocardiogram is just the fact that compared to, let's say, a chest X-ray or a CT, where it's relatively fixed position, where you're taking a scan, it's in one particular place, and then you're then analyzing that. Something like echocardiogram and any sort of ultrasound scan, it's going to be a lot of mobility in terms of the exact angle that you're using, the exact position that you're using. So that's something that needs to be taken into account when you're analyzing these images. Fine, so they're saying about the importance of echocardiogram, what it's used for, why machine learning and deep learning in particular is well suited for the analysis. They used 2.6 million echocardiogram images. So in this case, they will have used, well, it looks like they've used still images of from echocardiograms, which introduces this challenge of exactly what is the angle that's being used. Although there are certain particular views that you take when you're doing an echocardiogram. So for example, there's the apical 4-chamber view that they've mentioned here. And I guess the idea is that there's a standardization there in terms of the type of image that you're taking, but there's still going to be a degree of variation in terms of the exact position of the probe. Okay, they talk about predicting function as well, because what we saw in the abstract there was structural things, so identifying left ventricular hypertrophy, identifying left atrial dilatation. Let's see what they say about function. So ejection fraction is one of the key things you always see on echo reports. So they predicted the volumes in systolic and end diastolic. Okay. So it's also able to predict ejection fraction with reasonable accuracy also. Yeah, moderate performance. Okay, and the other thing they also did was predict risk factors. So did they do this based on the echo? Okay, so they predicted age, sex, weight, and height. Yeah, interesting. So this is another example of something that for me is a really exciting area of deep learning, which is where the machine actually is able to do things that humans can't. So here, for example, human interpreters can't look at purely echocardiograms and predict the phenotypes such as age, sex, weight, and height. Whereas what they're saying here is actually that their model, this EchoNet that they trained, is able to predict those purely based on the images. Another example of this was with retinal scans. So if you look at a retinal scan, someone trained an algorithm that can predict the sex of somebody, or say whether they think they're male or female based on the appearance of the retina, which again, it's not something that humans can do. So maybe there's some sort of features that the network is able to pick up here that is relevant. So that's interesting. And this is their ability to do that. Fine, and then a bit of discussion. Black box. Fine, so they're just talking about what they did. So previous deep learning work looked at obtaining the correct view and handcrafted scenarios with closely curated patient populations. Okay, so this is a fair point to make, that at the moment it does not surpass human assessments, and really the benchmark, or one of the benchmarks for looking at a machine learning model and whether you're going to be able to use that and incorporate that in clinical practice is the extent to which they can match or improve on human performance, because then you can start making an argument that maybe it should be incorporated alongside or instead of humans, although there's a big step from alongside to instead of. And here they're saying, so at the moment they're not surpassing human performance. Fine, recent. Approach of using types. Okay, I don't want to spend too long on this. Reproduce on human. Fine. Okay, so that's interesting. And a bit more details there, and the code is available. That's cool. Always good to see. So the code is actually available on GitHub here. and you can request the data. So you could request the data and retrain the model. Okay, next paper. How are we doing for time? 13 minutes, and we're still on week 1. So really I should be finishing this. Okay, we've got one more, but I'll have to speed up a little bit after this one. Detecting early infarction in acute stroke with non-contrast enhanced CT. And we don't appear to have access to the full text, or do we? No, we can't read the full paper, which actually will probably mean this is a bit quicker. Okay, so we're here again, it's going to be a CNN. We're trying to detect early infarction in acute stroke. So obviously with stroke you have this critical window for thrombolysis of less than 6 hours from the onset of symptoms. So anything you can do to detect this at an earlier stage is helpful. There's a company in London called Viz.ai who are also working in this area of trying to predict stroke at an early stage. And I wonder if this is... So this is a group in Switzerland, Canada, Korea. So it's a bit of a multi-international group. Okay, so we want to detect and quantitate infarction with non-contrast enhanced CT. Okay, so this is an interesting point here, is for all the patients that they included when they were training this algorithm, they had both the non-contrast enhanced CT images and a later MRI image within 1 hour. And that, I imagine, is what enabled them to do this study, is because they had the MRI data, which provided a higher granularity of data and was essentially the ground truth in this case, so saying exactly what was present on there, were there any infarcts, and they used that as the outcome, and then the non-contrast enhanced CT was the image that they then trained the network on. So they trained it to detect features on the non-contrast enhanced, which then was pointing to the presence or absence of infarct on that MRI, the high resolution scan. So they manually contoured them on the MRI, and you had the reference standard. So that's the the ground truth. Okay. And it found good agreements. The AI analysis of the CT agreed pretty well with the MRI. Cool. So that could be pretty helpful because it's harder to get access to an MRI machine typically in most hospitals, whereas CT is more readily available. So if you're trying to identify stroke and the size of stroke in an early stage, being able to do that on a CT scan with non-contrast enhanced can be particularly useful. And it looks like that this is doing a reasonable job of doing that. Okay, so that is the first week of February. Let's move on to the second week of February. What week are we on? I think we're on... Is it week 39 is the next one? Yes, week 39. We're on 17 minutes. Fine, so 9 papers. I'm going to have to be quick for this one. So we've got a computer-aided design electronic, I think, system to improve adenoma yield. So this is part of these ongoing studies that are coming out, I think, of the same group, Wang et al., who are doing various work looking at detecting polyps on colonoscopy, and there's been quite a few different previous studies published at an earlier stage on this. Then we have a real-time computer-aided system to detect colorectal adenomas. So is this again on... Yes, I think this is maybe just doing a similar sort of thing of detecting lesions on endoscopy, which is an interesting clinical problem that AI can help with, and it's another study. So we'll have a look at those two and see what there's difference between them. but an error to detect breast cancer on mammograms and explain whether it can be improved the performance of radiologist breast cancer detection. Interesting. So the key thing there is how is it going to be incorporated into this breast cancer detection pipeline. Machine learning models for tumor type based on sequence-based features and combination of features. So what type of features is this? What type of images are we talking about? Gradio Hub, a web platform for clinicians and biomedical research to try out models and test their reliability. Very interesting. So we want to decrease the level of expertise required to train and run machine learning models, so we can disseminate this to clinicians. Because ideally, we'll get to the point where clinicians and biomedical researchers who have never really worked with machine learning will be able to use these models and train useful things, because we just want to democratize access to that. That would be very useful. Machine learning models predict in-hospital mortality. Okay, fine. So there's quite a few models doing this sort of thing, are trying to predict mortality, predict outcomes of when people come into hospital. So interesting to read that one. Grading hip osteoarthritis on radiographs. Fine. Orbit framework, open source whole slide imaging analysis tool. So again, this sounds like it's again about democratizing access to this sort of thing. So if you have this open source software that people can then use, then histologists can use this just to facilitate their process and their way of analyzing scans. So the question there is, what sort of tools do they have, and what sort of tasks does that perform? And then DeepWAS, so this is going to be looking at genetic associations. Fine, so that one again is a little bit more outside my domain. Okay, so deep learning detection of polyps. What have they done? So it's a previously shown in non-blinded trials to improve detection, and I believe this was the same group, Wang et al. They've now done a double-blind randomized trial in China. Now, I'm interested in knowing, how did this work? So they have this AI that can flag up the lesions, and then they'll have the operator doing the colonoscopy. How do they double blind that? Okay, or a sham system. Fine, so it'll basically be a system. What do you mean by sham system? Is this where there's someone kind of behind it? Is it where there's a human flagging things up? By computer, okay, fine. Patients are also unaware of their own assignment. To achieve masking, the upper system was shown in a second monitor that was only visible. Yeah, okay, fine. So there's a second monitor that was only visible to an observer who was responsible for reporting the alerts, who presumably was not the endoscopist. Okay, interesting. So what I understand of this trial is that there was an endoscopist who was doing the study, doing the endoscopy. They were looking for lesions. Then there was another screen that an observer was watching, and if that screen flagged up there being a potential lesion, then that observer would say to the colonoscopist there was a lesion there flagged up by the system. And that system flagging it up, it was either their AI system, this CAD-E system that they'd created and have done previous papers on before, or it was this sham system. And I don't know what the sham system comprised of. Yeah, I'm just going to have a quick look to see if I can understand what the sham system is. It might be the case that I don't have access to this paper though. So let's see. Yeah, we're not going to see any more. Yeah, I'm not too sure what it will be. It could be the case that, would it be a human person looking at the polyps and saying something, or would it be somebody, or would it be a system that, I don't know, it just flags up randomly saying that there's a lesion? I'm not too sure. Okay, so then there's another paper. Was it in the same journal? Yes, Lancet, Gastroenterology and Hepatology. So same paper, a different group, I think. Let's just see if it is a different group. So this is those guys, and this one is a different group, I think. Yes. Where are these guys from? These guys are from Wuhan, Chengdu. So yeah, it seems to be a different group, but doing a similar sort of problem. So colonoscopy for detecting lesions, colorectal cancers. So they wanted to make a system. So maybe it's a bit different here. A quality improvement system to monitor real-time withdrawal speed and colonoscopy withdrawal speed, and to remind endoscopists of blind spots caused by endoscope slipping. So it sounds like this is doing a different sort of task. It's not just trying to look at the presence or absence of lesions, but it's giving some kind of feedback on the ways in which they're performing the task, and maybe trying to support their ability to pick up the lesions themselves. So again, endpoint was adenoma detection rates. So it did improve the adenoma yield as effective and safe. The question here is, how exactly did it work? Because we can see that it monitored the withdrawal speed and reminded endoscopists about blind spots. Okay, so is it a case that when there was a slipping and the scope moved, that then they would just remind the endoscopist and say, there's a blind spot here, have a look to see if there's any polyps there? It sounds to me like that's kind of what it was doing. So again, another tool that's supporting this, and you can envisage in the future having a system where you're doing a colonoscopy and you just have these different AI systems running in the background, feeding alerts and kind of advising you, and by doing that, improving the adenoma detection rates more broadly. Changes in cancer detection and false positive in mammography, a retrospective multi-reader study. Okay, I'm not going to spend too long on this one. Essentially, they trained an algorithm to look at a data set that previously been analysed by radiologists. And report better performance compared to radiologists. Okay, but they also did it with radiologists supported by the AI. The question is, what was the nature of support? So it sounds like in terms of performance, they had radiologists alone, AI alone, and then radiologists with AI, which makes sense. And as they've said, that supports this being used as a diagnostic support tool. What was the nature of combination? Was it a case that it kind of flagged things up? Or did it use the agreements and disagreements between the radiologist and the AI? It doesn't say. Let's have a quick look if we can get access to this paper. Yes, we can. So let's see, how does it work? Oh, it keeps on refreshing. Okay, so they tested it on a few different data sets, USA, UK and South Korea, which again is important to enable generalizability because of variation that you may have between countries, so you don't want to affect the performance. 26 minutes, so we're pressed on time. So let's see the methods. Talking about developing it, how did they then do the study? Here we are. So number of readers. Maybe I should move on to the next paper now, but the appendix. Fine, yeah, okay. So I don't know the exact nature in which this was combined, but I don't want to spend too long getting bogged down in this. Okay, deep learning system to classify primary and metastatic cancers using passenger mutation patterns. What is a passenger mutation pattern? This is within the genome sequence, so they had people who had the whole genome sequenced. Okay, so I think this is kind of bioinformatics task here. So we've got the tumor, we have the organ and the histopathology. Or no, they're saying that in general that's the strongest determinant, but sometimes there's not an obvious primary. Maybe we can use the genome sequence and analyze that. fine. So I think that they're analyzing the sequence here and trying to use passenger mutation patterns. Let's see what that is. I have not heard that before. Okay, passenger versus driver. This is going to be something to do with how active the okay, so a passenger mutation here we are. Driver mutation, it gives a selective advantage through either increasing its survival reproduction, and they tend to cause clonal expansion, whereas passenger, it doesn't have that effect. So it's more of a, they say, hitchhiker here. So, okay, what I understand of that is that basically it may be the case that, yeah, these are, we're using mutations here that are not actively contributing to the cancer being kind of adapted to its environment, but that are still present. So these passenger mutations, and using those to predict where the primary cancer is, I believe, the origin, which could be very useful. Okay, an online platform for interactive feedback in biomedical machine learning. GradioHub. Let's have a quick peek of what GradioHub is. Okay. I think I actually have already seen this. Yes, no, I did come across this. I had a little play around. Yeah, I mean, I think maybe this is a bit more of an educational thing. Definitely check this out if you're interested and have a play around. Basically, what is it again? So I think you can pick images, and it will classify that you give them a saliency map. Yeah, so essentially there's models here doing different sorts of tasks. You can upload an image of skin and it will suggest whether it thinks it's cancerous or not. So is there any saved history here that we can use? Still loading. Yeah, fine. So here someone's submitted the image of Pikachu. That's been classified as a malignant skin lesion. So it seems to be that fine. So it's a way for people to just try out models, essentially, and you can just test out data. Fine, and it can be fun to play around with also. 30 minutes. We've got quite a few papers to get through. Let's get cracking on these. Prospective and external validation of a machine learning model to predict in-hospital mortality rates. Okay, so again, there's a lot of these predictive models. Very interesting because this is what machine learning is good at. It's good at looking at lots and lots of variables, looking at lots and lots of data, and identifying different patterns. So here the outcome is in-hospital mortality rates, and what sort of variables they're looking at. They are looking at things from electronic health records. Does it say what data they're using? No, I don't think it says it in the abstract. Interesting. What are we looking at? Let's see, medals, methods. Okay, fine. So basically it's lifting, I'm just going to close these tabs, it's lifting things from the electronic health record and using that to predict mortality, and it's getting pretty good AUCs. Fine. So there's quite a few papers in doing this sort of thing. Again, this is something else that I've definitely seen before, of CNNs analysing radiographs. I've seen a paper a few years ago to detect hip fractures. Here we're looking at hip osteoarthritis. It makes sense. We know the features of osteoarthritis, of which I remember being taught 4 main features: osteophytes, narrow joint space, sclerosis of the joint, or like a thickening of the edge of the joints, and there's a 4th feature I can't remember right now. So it's feasible you can train a CNN to look for those features. So joint space narrowing, osteophytes. Okay. So there's another kind of task, a specific narrow task that an AI could do well at, which is analysing the presence or absence of osteoarthritis and grading it. Then we've got an open source whole slide imaging analysis tool. So let's see, what does the tool do? And then we've got this GUS one I'll talk about in a second. So what does it let us do? Let's look at some of the images. It's always quite helpful. It's taking a minute to load. I don't know why this is being so slow. So what's this saying? Is there some sort of automatic segmentation? So you can segment, presumably you can do some automatic segmentation, and then you can label those objects as being a particular type of structure. I don't want to wait around for the rest of these images. Fine, so it seems that there's some degree of auto-segmentation. And I guess essentially it will just be a case of doing the types of things that histopathologists like to do, so like to label structures, label the sizes, measure the sizes, that sort of thing. Classifying pixels, annotations. Fine. And then finally this GWAS one, identifying variants. Deep WAS, again, nice name. Just put deep on the front of anything. Fine. A little bit outside my area of expertise. Let's move on to the next week quickly because I'm going too slow. I think this is week 40 now. Yes, week 40, good. Oh, 11 papers. Yeah, I'm pushing myself to get this done in under an hour. Okay, I'm actually just going to look at the abstracts, save a bit of time. Deep learning robotic guidance for autonomic vascular access. Oh yeah, I did actually see this paper before. Pretty cool, to be honest. So looking at using AI to guide medical robots to access veins. I think it's automatic cannulation, isn't it? Robotic cannulation. So how do they do it? It's a portable robotic device. I'm going to Google to see if I can get an image of this device in a minute. But robotic cannulation, series of deep CNNs which encode spatial temporal information for multimodal imaging and real-time servoing. To use feedback information to move the robots. Okay, interesting. I mean, this, in my head, this would be like the future if robots are cannulating. That'd be pretty crazy. If they can do it safely and everything. That would be pretty mad. Yeah, because to be fair, we don't tend to use ultrasound when we're cannulating. If you had a robot that had that as part of it, you might be able to do pretty well. Okay, wow. I mean, would you be comfortable to have this on your arm? I don't know how I would feel about that. So the first step is segmenting the vessel, selecting the site, the vessel you want to go into. Another network to classify the vessel, I guess we're saying vein or artery, because you don't want to be cannulating an artery, and then cannulating and drawing the blood. Wow, I mean, I guess this must be an early stage, and I'm not sure how comfortable people would be about having this machine on them. At least at the moment. Maybe there'll be a bit of a paradigm shift. It's also quite big and bulky, so maybe we would want to have something that looks a little bit more streamlined, but obviously this is in development at the moment, so once you get the point where you can do this and the robot can technically achieve it, then you start looking at making it pretty. But yeah, that's pretty mad. Wow. Okay. Just going to make a note of that paper because I'm going to come back and read that in a bit more detail, look a bit more into that. Okay, electroencephalographic signature predicts antidepressant response in major depression. Okay, interesting. So yeah, again, imaging of the brain, so like MRI and EEGs as well, measuring the electrical activity, can be useful for predicting the development of different mental health disorders, depression, schizophrenia, that sort of thing. Then also doing prognostication. And here we're talking about the response to drugs. So it's another way in which it could potentially be useful. And they've looked to train, presumably, a deep learning model to look at response to sertraline. Okay, interesting. Yeah, so that's basically what they've done in Nature Biotechnology. What kind of model did they use? Oh, yeah, we can't access it. I don't know. Did they use an RNN or a CNN? Maybe it's a bit academic, but it's interesting doing that. Deep learning based prediction of late age macular degeneration. So what are we using here? We're using retinal images, and genetic variants. Okay, nice. So this is an example of a paper where they're combining different domains. So you're having this imaging domain where you're feeding in the image, and then you're also having the domain of genetic information. So doing some sort of genetic analysis and then ensembling those different methods. Network tree for the prediction of protein binding. Fine. Again, I'm going to skip that one. I mean, this is an important thing, is being able to predict what proteins are going to interact, because that can help you predict drug side effects. It can help you just understand the purposes of proteins and the functions that they have in the body. But I don't know too much about it. And again, we've got predicting drug protein interaction. Yeah, I'm going to skip that one. Deep learning of circulating tumor cells. What can we learn from the circulating tumor cells? If you find these in the blood of cancer patients, it can be a biomarker for precision medicine. Yes, it's feasible that you have a tumor somewhere in your body, and then some of these cells then enter the blood, and analyzing those might give you some information about the genetic makeup of the cancer that the patient has, and therefore maybe what types of treatments you have, or the prognosis, or different elements of information that are useful about the tumor. So auto-encoding CNNs with advanced visualization techniques. A fluorescent image of blood samples. Okay, so here they're looking at the number of circulating tumor cells, as I understand. So they're looking at blood samples. They are using fluorescent dyes to flag up the CTCs, the circulating tumor cells, and finding the counts, which is important for predicting survival. So they're not going down the route I talked about of this genetic analysis of those cells, which I guess maybe it's not necessary, because if there's a tumor and you can get a sample from that tumor itself, then you can do the genetic analysis on that tumor. You don't need to use a circulating tumor cell for that. Although perhaps, if that is a possibility, I don't know if it is, to be honest, but you could overcome the need for invasive biopsies, let's say, if you can analyze these cells peripherally, so you can take a blood sample rather than a biopsy, which is less invasive. They also found different subclasses of objects. What sort of subclasses? So subclasses of the cell types. A segmentation-free deep learning algorithm for diagnosing glaucoma on OCT. So they're discriminating glaucomatous I've actually not heard that word before. Glaucomatous from normal eyes without segmentation lines. So I have seen previous papers of trying to predict glaucoma, but maybe they use segmentation, so that's the difference here, is that they didn't segment it. To be honest, I'm not sure what the ins and outs of those different mechanisms are. We perform better than the conventional thickness parameters for diagnosing glaucoma. Fine, so is it the case that they didn't segment out the image of the OCT scan? They didn't segment that image and then analyze the segmentations. They just analyzed the image on the whole, I think. Prognostic significance of quantitative myocardial perfusion. So you have these stress MRs. Fine, so you can automate the analysis of that. Fine. So here we've got a thought, like an editorial, translating preclinical models to humans. Oh wow, there's a lot of papers published in this week. Translating preclinical models to humans. Can I see the full text? No, I cannot. Published in Science. So generalizing results from animal models to human models is a big challenge. And it's one of the reasons there's a lot of failures. Yeah, I think it's something in the region of 12% success rate. So 88% of things that get all the way through animal models fail in human models. Okay, humanizing. We may improve our success by humanizing computational models derived from animal experiments. I'm interested to understand what that involves. But it's feasible that that's like an alternative approach. It's evident to humanize. I want to read that article. I'm actually going to make a note of that and read that later. I don't think I'm going to make it for 1 hour. I'm on 46 minutes and I've got like 1 or 2 weeks left. So machine learning for active matter. What do we mean active matter data? Images and track objects. So talking about images where things are moving. I want a little bit more context on this. What sorts of things are we talking about? Active matter. Fine, what, like biological systems that are moving while they're being analysed? Okay, fine. So this is, again, it's quite pure research focused, perhaps? Fine. And then AI in endoscopy. So we saw those 2 papers in endoscopy, I think, the previous week. So this editorial is probably commenting on those, and they were both published in the Lancet, Gastroenterology and Hepatology. So here, I can't read it, but essentially, yeah, fine. So it has potential to improve interpretation of images and improve the detection rates. Let's now move into the second half of February. I think, are we on week 40 or week 41? I think it's 41. Yeah, 41. Okay, 4 papers. Deep learning approach to antibiotic discovery. So did I see this paper or a similar one? Yes, I think I saw this paper. So at the moment, as far as I'm aware, there are 4 reports of drugs that have been developed using deep learning and that have that basically accelerated parts of the drug acceleration pipeline. So I actually wrote an article a week or 2 ago about the use of artificial intelligence in drug discovery, which I will link in the description below. And essentially, one of the issues with drug discovery is the very high failure rate of drugs and the length of time that these different stages take. So in total, it takes about 15 years to go from the initial conception of a potential molecule up to it being developed. It costs around £1 billion or a bit more than £1 billion, and a lot of the drugs fail. It's a very, very low, I think it's 3% overall success rate, which is extremely low. So if you can use AI deep learning, then what that could enable is it could enable you to accelerate that process, because perhaps you can accelerate different stages within that pipeline, and you could also reduce the costs, because by accelerating it, by increasing the success rates, then you can save a lot of money and develop these new drugs. And there's a lot of money to be had and to be made here in making these new drugs, because there are so many different diseases and so many different drugs that could be developed that there's a lot of potential, and there's stacks of startups. In the article that I wrote, I mentioned that there's 212, the latest count of startups working in this area. There's 43, I think, pharmaceutical companies all working in this area of trying to use deep learning and AI to accelerate their ability to develop these drugs. And this is one of them. So I remember reading this before. So this is from Harvard. Was this in collaboration with a startup? I'm trying to remember. But I think these guys, essentially they might just be Harvard. I think they repurposed an antibiotic, or they repurposed something else as an antibiotic. So they trained it to predict antibiotics based on structure. But Halasana previously wasn't an antibiotic, if I remember correctly. Yeah, what is Halasana? This might just show some news on it. Okay, so originally this was research for the treatment of diseases, but development was not continued for the application due to poor results in testing. Because the way that they test it is by putting it through different sorts of tests to look at the how well it dissolves, the structural stability, the potential risk of side effects. So they obviously developed it up to a certain stage, then kind of dropped it, decided not to proceed with it, probably proceeded with an alternatives, but then that molecule will have been recorded on a database somewhere, which is accessible if you're doing this kind of analysis of deep learning. And this group have essentially found that and felt that it might have, or their models predicted that it might have some antibacterial activity, and they've tested it in mice and it has shown broad spectrum antibiotic activity. So this is super exciting, because here they might have found a new antibiotic by drug repurposing, and one of the nice things about drug repurposing is it cuts out a lot of the pipeline, because it's a drug that's already been developed, it's already been structured, the structure's been identified, and the molecule's been synthesized, and so you don't have to redo that aspect of things. And yeah, on the whole, it's pretty cool. So that's a lot easier than doing this kind of full pipeline. And in this case, it sounds like they didn't fully develop the drug and test it in humans previously, but or maybe they did. It depends what this kind of testing it was. But sometimes you can take a molecule that's already been developed and is now being used in humans, and you can repurpose that, and that essentially cuts out the whole pipeline, because you've already shown that it's safe in humans, or at least in particular subpopulations of humans, and now you can repurpose it and use it for something else. So that's what they did for this antibiotic, and it's very exciting. This could be a new antibiotic, and we have a big issue with antibiotic resistance at the moment. So any new antibiotics is fantastic. Cellular and molecular probing of intact human organs. Okay. Particularly challenging to render transparent. Interesting. So this is quite interesting. I don't know too much about this, to be honest, but it sounds like this is that they're talking about rendering organs. So you take an intact organ, you perform histology, and then you can look at it at a higher resolution. I don't fully understand that. In the interest of time, I'm not going to go deep into it. machine learning individualized treatment rule in first episode of schizophrenia. So this is similar to the paper that we saw before in depression, where we are looking at analyzing features of the schizophrenia. What kind of data do we use? So it's a bit different. In that paper, they used EEGs and tried to predict response to sertraline. Here we're looking at discharge diagnoses and the medications and ambulatory claims. Okay. Okay, and it recommends the best treatment. So I guess what I understand of this paper is that they analysed the outcomes from different people with schizophrenia over this 7-year period, and they looked at how well they responded to different antipsychotics and then made suggestions for individualized treatment rule for when people have a first episode of schizophrenia. So then when you're presented with a new patient, perhaps with the same features as the other patients, I'm not sure if that's the case here or not, then it will suggest what best medication to go with, and it seems like aripiprazole and amisulpride are the two that they recommend in this study. But as they point out, it's kind of early stages, but it could be the case that you can predict exactly who's going to respond best. And this is a really potentially interesting and useful area generally of machine learning in these kind of problems, because with a lot of the mental health problems, depression, schizophrenia, you essentially have a lot of different drugs that you can use, and at least from my experiences, sometimes you'll go with one particular drug and sometimes you go with another particular drug. It depends a little bit on the preference of the clinician and the experiences working with the different drugs. There are some guidelines, but often the guidelines are try this or try this or try this, and then second line, try this. So if you can actually predict that in a more individualized way, a more personalized way of, well, which medication is this patient going to respond to best, it's a great outcome, because when you start taking these medications, often you wait 6 weeks or a few months to see the effect of the drug. If you can predict that earlier and lead to a better predicted outcome and kind of better accuracy of predicting how well it's going to respond, then you're just going to improve the outcomes more broadly. So we're now on to the final week, which I think is week 42. Yes. And we've got 5 papers, we've got 4 minutes. I'm going to be just over the hour mark, so this is going to be a pretty long video. I don't know if anyone is still watching this video, but I'm going to keep on going. Okay, so 3 papers and then 3 primary papers, 2 prospective papers. Image, impact of a deep learning system on the histology classification of liver cancer. Fine, so it sounds like we're looking now at not the model itself, we're looking at what's the impact in a clinical setting. Because it's been established for quite a while that we can classify histopathology images pretty accurately in a lot of different conditions, such as in liver cancer. So here what they're saying is, yes, it continues to rival human performance, while their actual impact when they're incorporated into clinical workflows remains unexplored, and classically we see a decline in the performance when you go from a retrospective study of looking at existing data and then apply that model to new data in a prospective study. A classic example of this was a CAD, computer-aided detection system for breast cancer, where it was very, very superior in when initially applied looking at a retrospective data set, and then they did prospectively, they saw a massive drop from something in the region of 90-something% down to like 70-something%. And there's various reasons for this, which they probably took about in this paper. Yep, so they've got the system to evaluate the type of cancer. It has pretty good accuracy. And improved the accuracy of 9 pathologists who fell within well-defined experience levels. And it impacted the decisions of all of them. And it helped at all different levels. Okay, so basically the argument here is that yes, when you incorporate this particular model into a clinical setting, it still improves outcomes. So They show that it's accurate, that it works well, and then they say when you add that to a pathologist at any different level, whether they're a more junior pathologist or whether they're an experienced pathologist, it will still help them to improve their diagnostic decisions and improve the outcomes overall, which obviously is a good thing. And again, this, I think the reason for this is because it's very much a confined task that we're doing here. We're looking at histopathology slides and coming up with a diagnosis, and there's not as many variables outside that histopathology slide. It's a relatively contained environment. Chexpedition. Chexpert. I've not actually heard of this. I'm aware of ChexNet, which was this chest X-ray analysis AI that came out of Stanford, and it looks like this is the same group because there's a lot of familiar names there. We've seen recent advances in the application of deep learning to chest X-ray interpretation, which identify 3 major challenges for translating into a clinical setting. And we examine the performance of top-line performance of TB detection, pathology detection on photos, and pathology detection on data from an external institution. Okay, so Chexpert competition. So it sounds like this is a competition to see how well people can do on these tasks. And they perform or exceed radiologists. Okay, so again, here they're focusing on the clinical translation, because with these papers, with this ChexNet's work, it's very much like in an earlier stage. Let's see if I can access this paper because I'm quite interested in this one. And it's an archive, so I should be able to. Three major challenges. What are these 3 major challenges? That's what I want to see. Oh, do they mean the challenges in terms of these are the 3 types? Okay, fine. I thought they were going to talk about the technical challenges of analyzing chest X-rays of AI. Fine, okay, but that's interesting. So they're looking at translating this sort of task to these other problems. Non-invasive diagnosis of non-alcoholic fatty liver disease. So this is an interesting problem because non-alcoholic fatty liver disease is very prevalent within the population, and sometimes it leads to adverse outcomes. It increases the risk of hepatocellular carcinoma, for example. And so if you can detect it at an earlier stage, that's great. The gold standard has typically been biopsies, but as with any invasive procedure, biopsy has a risk of side effects. So therefore, it would be great if you can actually pick up the non-alcoholic fatty liver disease at an earlier stage with something non-invasive such as an ultrasound. Here they're talking about, they use MRI-deprived protein density fat as the reference. And are they talking about ultrasound? Yes, ultrasound data using one-dimensional CNN. So again, the challenge of CNNs being used on ultrasound is the fact that there's this variation in position of the ultrasound. So classifier to diagnose it and a fat fraction estimator. And how did they find they did? It looks like they did. The algorithm did pretty well. So it could be useful for the diagnosis. I mean, still there's a challenge of doing the ultrasounds and actually getting these things analysed with ultrasound. We've got -1 minute left to hit my hour target. And there's these 2 editorials. AI in cancer therapy, transforming global health with AI. AI may affect cancer therapy, drug discovery, which we've talked a bit about. These processes are expensive and time-consuming, variable treatment outcomes. So again, here, if you can predict the response to cancer therapy, that's great. I imagine you might talk about this. Yeah, so this is another thing to do with drug discovery is matching patients to clinical trials. So ideally, you want to find patients, like subpopulations that are going to respond well to your drugs, so that you have a maximum chance of successfully getting through clinical trials. And personalizing cancer therapy, yes, by being able to predict what the outcomes are going to be. And then transforming global health. It could revolutionize health and healthcare in low and income settings by addressing the large knowledge and judgment gaps. Some key challenges need to be addressed. I mean, that's doesn't give a lot of detail. Let's see what it says. Can we access it? Probably not if it's New England Journal. Fine, I mean, that's a pretty short abstract. Yeah, I guess a lot of these models have the potential to be used quite widely in third world and in developing countries. An example of that could be all the stuff that's been done with retinal scans and trying to diagnose retinopathy. If you can make that so that you can access it on a smartphone, then people have a lot more access to smartphones than they do to OCT scans, and they have more access to smartphones that can do this kind of analysis, if you have a good AI algorithm, than they do to ophthalmologists who can interpret those scans. So you can just disseminate the ability for people to analyze their retinas and the health of their retinas. And that's one example. Obviously, a lot of the things we've talked about essentially can just democratize access to these kinds of information, this kind of analysis, these kind of diagnosis and predictions and all that sort of thing, in ways that it's harder to do if you have much fewer doctors. Obviously, there's these issues with here. We're looking at kind of combining AI with clinicians and looking at how best to do that, and we don't feel super comfortable about replacing doctors with AI, quite rightly so in my opinion. Whereas in these countries where you have so many fewer doctors, you can make a stronger argument that actually an AI analysis is better than no analysis. So there's a strong argument for using AI, and there is a lot of potential for it to help improve global health if implemented well. Cool. So those are all the studies that were published in February that have been flagged by Dr. Penguin and that I'm going to be reading. To be honest, I'm not convinced that it's super valuable me recording myself talking through these papers. If you feel otherwise, let me know and I'll keep on doing it. But probably what I will do going forward is actually doing a bit more reading beforehand and then just summarizing the papers in a succinct way, so that then there's a resource for people who want to read these sorts of papers and understand what's going on in a sort of kind of condensed format, I guess, to save them having to then go and read all the papers in depth themselves. So probably I will try and shift a bit more towards that. I may still keep recording these. I'll have a think about it. As I said, let me know if you have preference either way, if you're interested in seeing these or less so. But yes, in terms of the interesting themes that were raised, we talked about drug discovery and the roles of AI in that. We talked also about ultrasound analysis, and it seems that there's quite a few papers looking at analysis of ultrasounds. So that's echocardiograms was one example, and also non-alcoholic fatty liver disease. We saw a general shift from the technical development of these algorithms into clinical application. So we saw several papers that were looking at the performance and the improvement in health outcomes once you implement it clinically. So we saw the colonoscopy paper that did that. We saw the breast cancer screening paper that did that. We saw the histopathology slide analysis that did that. So, and the chest x-ray is one. This Chexpert competition, I think, was also looking at clinical outcomes. That's a key development within this area of research applying AI to healthcare. We've seen a lot of success in narrow domains over the last 3, 4, 5 years. Now we need to move more towards looking at the impact clinically when you put it into the clinical setting, into the clinical workflows, and see, does it lead to an improvement in outcomes or not? Because it's definitely feasible that they wouldn't lead to an improvement in outcomes. So it's important that we do validate that we do that prospectively, that we do that in multiple different populations, that we ensure there's generalizability of the algorithms, and that we generally just make sure that actually these algorithms are improving outcomes when applied to a clinical setting. We should also be on the lookout for potential side effects or potential negative consequences of these algorithms, and make sure that there's no harm that's being come about from the algorithms, or there's no unforeseen side effects. And the purpose of these studies is to help us to elucidate whether there is any of that. But yeah, that's everything for today's video. This is still quite experimental, so let me know what you think of this format in the comments below, and I will decide exactly how I'm going to do this in future. I think this probably will be the last one where I go through it in this way and just record it. It's fun for me, and I'm going to be doing it anyway, but to be honest, I'm not convinced that it's super valuable and super good use of your time. So I probably will shift more towards an approach where I'm prepared in advance and talk through the papers in a bit more depth. But that being said, I hope you enjoyed the video. Leave a thumbs up, leave a comment below if you did like it, and I will see you in the next video.